Welcome everyone to the Stanford Cancer Institute's Breakthroughs in Cancer Seminar Series. We're very excited today to have Dr. Hong from MD Anderson, and happy to have everybody here in person and all the folks online. And now it's my pleasure to formally introduce our speaker, Dr. David Hong, who really has an incredibly impressive biography, and for the sake of time, I'm going to keep it to the highlights. Dr. Hong is a Douglas E. Johnson Endowed Professor and Deputy Chair of the Department of Investigational Cancer Therapeutics, which is the Phase 1 program of the University of Texas MD Anderson Cancer Center. It also serves as the Clinical Medical Director of its Clinical Translational Research Center. He has been a pioneering driver and leader in forming one of the most largest, highest volume, most really innovative Phase 1 clinical trial programs in the world. Thousands of patients enrolled on clinical trials every year, hundreds of active studies, personally been the principal investigator of over 100 research protocols supported by an array of institutions, including the NCI, CTEP, and industry. Over 400 publications in leading cancer journals. And as a drug developer, I think your mark is really made by the drugs you help shepherd to FDA approval and to the clinic for our patients, and has led the early development of a remarkable array of drugs, including cabozantinib, dabrafenib, trametinib, regorafenib, lenvatinib, sotorasib, most recently, and then when he'll tell us about his experience kind of leading and building his KRAS portfolio. Also founded multiple companies and an expert on multiple oncologic targets, including c-Met, NTRK, and adoptive cellular therapy. He is the recipient of numerous awards, including the Jesse Jones Award, Best Boss Award at MD Anderson, the Erwin Karkoff Award for Clinical Research, and also is well known as a mentor to junior faculty and fellows, which has been recognized as Mentor of the Year Award at MD Anderson, the Entrepreneurship Award in the Division of Cancer Medicine, and just a remarkable track record of folks and trainees who have gone on to make their own impact in the world of cancer and early drug development. And with that, Dr. David Hong.
Thank you, guys. I want to say I'm just really honored to be here. Stanford has always been kind of like the coolest place on the planet, kind of place that you always want to visit. When I got the invitation from, I think it was Anthony, it said something like, this is a great seminar. You have speakers like Jennifer Doudna, Nobel laureate, founder of CRISPR. No pressure, no pressure, right? And I know the hospitality was just great. I got to meet a number of your faculty. Clearly you guys were just generous, because obviously you guys were in Game 4 of the Warriors versus Lakers, and they dismissed that game to take me out to dinner. So I just want to thank you guys for taking me out to dinner last night. It was a close game, wasn't it? Sorry about that. Okay, let me start out with this. So it is an incredibly exciting time. Incredibly exciting time in oncology drug development. I don't see George here. Is George here? George is here just a second. George was telling us last night how he was one of the first, I think, graduate students in the Department of Molecular Biology here at Stanford, almost 40 years ago. That's a long time, George. We were like, wow, that's a long time ago. But it has taken 40 years for us to get to this point. And many of you I know are probably in the lab, but what I can tell you is all this sweat and tears that you have done, all the discoveries that were created 40 years ago and even now is now coming to fruition in our cancer patients. These drugs are beginning to work and to help these cancer patients, who many of them had no other opportunities. So I'm going to talk to you about KRAS, or RAS in general, one of, I would argue, the Mount Everest of oncological oncogenes that occurs in close to 40% of all cancer patients. And I'm going to talk about the past of how we tried to target, what is happening currently, and I think what will really happen in the near future. So these are my many disclosures. I'm sorry I have so many disclosures. So the objective of today's talk is really to review some of the discovery of RAS, what happened in the lab, and its relevance in cancer. I cannot go over, obviously, all of the history of RAS discovery and the details because it would just take us too long. But I will talk really about specific findings that eventually led to therapies that we tried in the past. We will talk about the past attempts at targeting RAS, and then I'll talk about the current clinical landscape of these new inhibitors or G12C inhibitors that are now impacting patients. Lastly, I'm going to talk about really the incredible promising future steps in targeting this gene. So, talking about another technology, this is rocket technology, and we kind of stopped at Saturn V in 1968. And a Texas transplant from, California transplant from Texas to Texas, really with his company SpaceX transformed this, and it took them almost 40 years. And Falcon 9, right, really transformed the space technology and transformed this whole industry. It has taken us about 40 years to get to this point now in RAS. If you guys recall, RAS is really divided into 3 isoforms, H, N, and K. And within that, there are certain codons, 12 being the most common, 13 and 61. And when you break this down, you see that there are different mutant alleles in different tumor types. Pancreatic cancer is the most is the tumor type with the most amount of KRAS mutations, 91%. And many of you know that this is a deadly, deadly disease, that even now, current chemotherapy has limited efficacy. Colorectal cancer, 68% of patients have KRAS G12, some kind of KRAS mutation. And lung, as you can see here, close to a number of these patients will have some kind of KRAS mutations, the most common being G12C, and we'll talk more about that. Melanoma is also, although BRAF is very common, can also harbor specific RAS mutations, particularly Q61. This diagram is really a simplified diagram of the activation of oncogenic RAS. And this has taken over 60 years to build, right? To understand all of the nuances of this. And there's probably much more that's laid out in this very simple diagram. But in a sense, it starts out with RAS as an oncogene that needs to be post-translational modified at the C-terminus through a process called farnesylation. This then allows RAS to bind to the inner plasma membrane. RAS exists in both an on and off state. It is a GTPase. And RAS, through what are called guanine exchange factors, like SOS, is then turned on through GTP, and then signals downstream of other molecules such as BRAF and PI3 kinase. So, again, I cannot go into all of the details of the discoveries of this very complex pathway, but let me start out with the first one, and that was the first discovery of the actual RAS gene in mice. And this was conducted in 1964 by Jennifer Harvey, who discovered that there was a virus called monoleukemogenic virus, and that this virus that could lead to leukemia at some point would recombinate with another gene within the rat called rat sarcoma gene. They didn't know exactly the details of it at that time, but it recombinated, often leading to sarcomas in these animals. Later, Kristen and Meyer discovered a similar gene that recombinated also with a virus that led to the discovery of KRAS. Now, it was almost 20 years later until in 1982, and I would say 1982 was really a pivotal year in RAS biology. And essentially, 3 independent labs, Dr. Barbacid, who is at Cold Spring Harbor, Dr. Weinberg, who is at MIT, and I think Dana-Farber, and Dr. Channing Dur's lab at the NCI identified and cloned the actual HRAS genes in human cancer cells. This eventually led to the discovery of KRAS and NRAS, and ultimately discovery that certain mutations, specifically in codon 12, can lead to oncogenesis. I think if RAS is ever nominated for the Nobel Prize, these three and Dr. Kevin Choquette, who discovered the first inhibitors of KRAS G12C, will ultimately win the Nobel Prize for this. But this was a seminal period in the development of RAS, but it would take another almost 40 years, right, for us to discover direct inhibitors of KRAS. One important finding was this discovery that post-translational modification at the C-terminus occurred through farnesylation. And this was also led by Dr. Dur and others. And this led to kind of this explosion in development of farnesyltransferase inhibitors in the late 1990s and early 2000s. This is a very small slide. You cannot see all the details. And the reason is that these are all of the trials, farnesyltransferase inhibitors, that were conducted, and they were all negative. And this threw a cold water on this whole field of RAS drug development. We would discover sometime later the reason for that was because there was an alternative way of post-translation modification. That was called granulation. But at the time when they had developed these farnesyltransferase inhibitors, that was not known. It turns out, this was actually one of my first studies that I ever did in 2005. This was an NCI study of tipifarnib and sorafenib. And we thought maybe targeting RAS plus downstream BRAF, we could hopefully lead to RAS inhibition. It did not work in RAS tumors, but it seemed to have some efficacy in medullary thyroid cancer patients with RET mutations. And it's because, at that time, which was not really fully understood, sorafenib also blocked RET. It turns out now we have a better understanding of RAS. In fact, we now know that HRAS is not granulated. It's only farnesylated. And if you look here, tipifarnib has had a resurgence. It is working in patients with HRAS head and neck and HRAS urothelial cancer. Okay. Probably the last significant finding in this RAS story is this really kind of the RAS understanding of the downstream pathways downstream of RAS. And there are many, many, many investigators, some of you who are involved in understanding this. But unfortunately, although we now have many drugs, such as BRAF inhibitors, several BRAF inhibitors, several MEK and ERK inhibitors, this has led to approval in BRAF mutated cancers and also NF1 mutant cancers, but none of them have actually led to any specific approval in RAS mutant tumors. And why is that? It's not entirely clear. We think some of that is just lack of inhibition, but likely also there seems to be a very low therapeutic window for these. In other words, these drugs, particularly in combination, seem to be very toxic, right? And this is one example of an ERK inhibitor that was conducted by Dr. Channing Dur's group out of UNC. Okay, so, and so I think before 2012, I think many people, particularly in industry, thought there's no way we're going to be able to target this thing, RAS, even though it's present in 40% of cancer patients. Why? Because just structurally, it has a very smooth surface at limited binding sites. It has a very tight intracellular binding region to the GTPase. And other attempts, whether it's farnesylation or downstream inhibition, just did not seem to work. But we had a SpaceX moment, a Falcon 9 moment, when in this paper in Nature in November 2013, Dr. Kevin Choquette and his group showed that a molecule could target what's called the allosteric pocket, the switch II pocket near the GTPase pocket. in a way binding irreversibly, covalently to the cysteine 12 residue, locking KRAS. Remember I said it exists in this on and off state, locking it in this off state. Well, how prevalent is the G12C mutant allele? It turns out it is pretty prevalent. In fact, if you look at ACR gene in non-small cell lung cancer, it's about 13% of all non-small cell lung cancers. In other tumor types, it's not as prevalent, but still prevalent, right? Like in colorectal, 3 to 5%. In pancreatic, unfortunately, it's only about 1 to 2%. But it also is present in other tumor types like endometrial. We looked at this in our database, in our large metastatic database, and it was fell out pretty much aligned with AACR gene. So I had the honor of leading this phase 1 trial, the first KRAS G12C in the clinic, and this was a trial that one of the fellows asked me, how do we get trials? To be honest with you, it was really through serendipity. One of my fellows, our phase 1 fellow, was actually, I had gotten him a job at Amgen, and eventually he became the clinical lead for sotorasib and called me up and said, Dr. Hong, would you like to lead the first KRAS inhibitor trial? I was not a RAS expert at the time, but he came and asked me if I wanted to lead it, and I said, absolutely, I would love to see if we can get this trial up and running for you. This was a standard trial. It was 4 dose levels, and then once we reached an MTD, or recommended phase 2 dose, we expanded in 3 subsets: non-small cell lung, colorectal, and all solid tumor types. So as a phase 1 doctor, a doctor who, my main job is to see what are the toxicities that we see with these new drugs, and it was incredibly well tolerated. The main side effects that we saw were diarrhea and a little bit of nausea. We did see some grade 3 elevations in both the liver function tests, but there were no DLTs observed even at the highest dose, what are called dose-limiting toxicities. No deaths related to the drug, and only 1 patient was discontinued because of a side effect. And we began seeing responses I had never seen before with any other inhibitor in RAS patients. And we started seeing responses, and the overall response rate in non-small cell lung cancer was close to 32%. And you can see here the waterfall graph. In colorectal, however, the response rate, we did see a handful of responses, but it was much lower than that was in the non-small cell lung. And that told us we had learned a similar story from the BRAF story. But we kept that in the back. We started to understand how this may work. This led to the phase 2 trial. It was actually a phase 1, phase 2 trial, and I ran the phase 1. And then after we ran the phase 1, I literally handed it over the trial over to my colleague, Dr. Skolidis, in our lung cancer group. And many of these studies, as you know, right now in oncology are that. They transition from phase 1 and then almost lead directly into phase 2. And this was led by Dr. Skolidis, and his team did a great job. And you can see here the response rates were even better than we initially saw it in the phase 1. It was a 37% response rate. The progression-free survival in this very heavily pretreated chemo-refractory of non-small cell lung cancer patients was close to 6.8 months. This led to the first ever FDA approval, accelerated approval, of a KRAS inhibitor in any tumor type in non-small cell lung cancer. And so from the time in 1982 to this approval here, it was almost 40 years until the discovery of this, leading to this approval. This eventually led to now in the second line setting, and this was a study that our colleagues at Sarah Cannon, Dr. Johnson, presented at ESMO last year, looking at second line sotorasib compared to standard care docetaxel, and it did meet its endpoint, progression-free survival of 5.6 months, a little bit lower than I anticipated, but it did meet its endpoint, and will eventually lead to the approval in the second line setting. Right behind the heels of sotorasib is this other drug called adagrasib. And adagrasib is made by a company called Mirati. I think Mirati's based, no, they're not based out of San Francisco, they're based out of San Diego. But Mirati, I can tell you, the only difference between this drug is really its pharmacokinetic or PK characteristics. It is like sotorasib, it's a covalently binding allosteric inhibitor of that switch II pocket, locking RAS in that inactive state. It too showed significant responses. It had actually a higher response rate than sotorasib, close to 43% response rate, with a progression-free survival of 6.5 months. And you know, a lot of people mentioned that the response rates were higher than adagrasib, but when you looked out at other characteristics such as duration of response, progression-free survival, and overall survival, essentially the drugs appear to be equivalent. One aspect that all lung doctors here, I don't know if there's any lung doctors here, but is important is how brain penetrant are these drugs. We think that both drugs are likely brain penetrant, and this is some data both from adagrasib and sotorasib showing that in treated brain mets, since most of these trials did not allow for unstable brain mets, these patients do seem to have some effect on CNS metastases. So what about this story of why are we seeing lower response rates in other tumors? So in colorectal, you can see here sotorasib had about a 12% response rate and adagrasib had about a 19%. I'm going to tell you a little bit about what we know, again, from the BRAF story later down the line. But we did see this in pancreatic cancer. And the response rates here were not dramatic. They were about 22%. But I think the reason that this paper got into New England Journal was not necessarily by the response rate, the efficacy of this, but the fact that this was proof of concept that KRAS could truly be targeted in pancreatic cancer with a direct inhibitor. And we saw definitely clinical benefit in these patients. Many of these were very highly refractory patients who had multiple lines of chemotherapy and even on other trial drugs, but yet were benefiting from these direct inhibitors. This is adagrasib, had close to a 33% response rate, and biliary cancers also had a high response rate. One question I always get is, can these G12C inhibitors be tumor agnostic? Can they be tumor agnostic? Can we get tumor agnostic approval like we have with larotrectinib, RET kinase inhibitors, et cetera? It's hard to say right now because the numbers are small. But we have seen, as I shared with you, multiple responses in pancreatic cancer, but also we've seen now multiple responses across a number of tumor types, including endometrial, other diseases that harbor rare alterations of KRAS G12C. This is an old slide, but as soon as sotorasib was approved, there was a rush of new G12C inhibitors into the clinic. But this drug development business is not easy. In fact, I remember I was at an SAB with Janssen in 2012, and they told me they think that they had a KRAS inhibitor. However, it never left the preclinical stage because they developed some kind of toxicity that they were too afraid to bring into the clinic. Likewise, Lilly had a molecule, their first generation KRAS G12C inhibitor, had some off-target effects that were in the phase 1 that ultimately led to the death of that molecule. They now have a second generation molecule, which they recently reported at AACR. But ultimately, who will win the Iron Throne is hard to say. Sotorasib was the first drug to be approved. Adagrasib now also has approval. My guess is that there will be other G12C inhibitors and even better inhibitors that will eventually come into the fray. So are we done now? We've got, I would say at this point, 10 years ago, we were not even at the base camp. We are now at the base camp of this mountain called KRAS. But we have far, far to go to actually reach that summit. Because the response rates, although they are significant and clinically beneficial, are not dramatic. One of the questions is the development of resistance. Most small molecules we know will eventually develop resistance. And I laid this out in a review in cancer discovery with one of my fellows of the different preclinical data that we now have, both on resistance and overcoming resistance. And Dr. Mark Awad from Dana-Farber beat us to the punch because he was able to get this data out. We had all these resistant samples and we were analyzing it, and then he published this in the New England Journal. And in this paper, he laid out 15 patients, many of them colorectal, many of them non-small cell lung. And as you can see, it was somewhat of both a wonderful paper, but also somewhat disappointing because there was not one alteration, right, that we could argue that the majority of these patients developed. In fact, if you look at across these different patients, each row is a different patient, you can see many of them had multiple alterations and multiple mechanisms of resistance. And they fell into these different categories: mutations at that switch II pocket that prevented the binding of the drug, or bypass signaling, whether it was through ERK or BRAF or PI3 kinase, or even development of fusions, new fusions that led to continued growth of the tumor, mutations in downstream effectors such as RAF, et cetera. And lastly, state transformations, which we sometimes see in non-small cell lung cancer, going from non-small cell lung to small cell and to squamous cell. We looked also, we did a larger data set with our patients than the sotorasib non-small cell lung cancer study, and we did see that unfortunately it was not just one mutations, but multiple mutations that most of the majority of these patients had, and they spanned everything from PI3 kinase to other alterations such as FGFR. Likewise, in our colorectal data set, again, multiple different resistant mutations. Many of them in this data set where we combined this with panitumumab, we saw patients actually had upregulation of KRAS, actually of the wild type, suggesting that may be also a mechanism of resistance. We now know that only about 40% of these patients actually respond. Why doesn't the rest of the 60% respond? There were early hints from the phase 1 data sets that we had in the biomarker that there were co-alterations that could lead to decreased efficacy of these drugs, particularly, as you can see here, KEAP1 and STK11. My colleague, Dr. Skolidis, a number of investigators throughout the country, myself, et cetera, got together and we tried to pool as many G12C patients that we could gather to understand whether or not co-alterations eventually affected the efficacy of KRAS G12C. And lo and behold, we did confirm that this was close to over 480-some patients. When we looked at their overall co-alterations and their survival curves, we found that KEAP1, SMARCA4, and CDKN2A were co-alterations that definitely affected whether or not these drugs actually had significant impact in these patients. STK11 turned out, although it affected response, ultimately did not pan out in the context of survival and PFS and overall survival. This has already led to a number of thoughts of how do we increase the efficacy of the single agent, right? Can we block downstream effectors such as BRAF or MEK? Can we inhibit other GEFs such as SHP2 or SOS in combination? And I've been involved in both the CodeBreak 101 and the Crystal-1 combination trials. And one lesson we can learn is actually a lesson from BRAF. So when vemurafenib, the first kind of really dedicated BRAF inhibitor, was out there, I don't know if you've had Keith Flaherty here to talk about his role in BRAF. He will tell you, preclinically, he thought this thing was going to work in colorectal cancer. It turned out it didn't work in colorectal cancer. It worked in melanoma, right? And it only had a less than 5% response in colorectal cancer. And this paper that came out from Prahlad et al. In Nature of 2012 suggested the reason for that was because there was an EGFR feedback loop. So I got with Scott Kopetz, who's a colleague of mine at MD Anderson. I said, Scott, let's design a phase 1 trial to test this hypothesis. The story behind how we got this trial up and running is a long story, but long short of it, we did a trial of them, irinotecan, and cetuximab, and showed indeed that was the case. In fact, the response rates went up to close to 40%. This led Scott to pitch the idea of a SWOG phase 2 of the combination compared to standard of care irinotecan and cetuximab, and showed that indeed it improved PFS and overall survival. This led to the development of what are called the Beacon studies, encorafenib, which was a second-generation BRAF inhibitor with cetuximab, and Dr. Kopetz led those studies and eventually led to the approval of encorafenib and cetuximab in colorectal cancer. We knew that this story was likely going to recapitulate with KRAS, and this group from Italy unfortunately beat us to the punch and published this preclinical data suggesting that indeed EGFR blockade reversed KRAS G12C inhibition in colorectal cancer. This has led to these combinations now with KRAS G12C. As you know, remember the response rates were close to 7 to 12%. In combination with either panitumumab or cetuximab, we've been able to increase the response rates to 30 and 46%. A second generation molecule from Genentech called GDC was recently reported at AACR in combination with cetuximab in KRAS G12C colorectal cancer. The response rates are now close to 66%. This has led to Crystal 10, which Scott is leading. And like I said, it always benefits if you have a phase 1 that eventually leads to a phase 2, because now Scott is leading this Crystal 10 study, and hopefully this will also lead to approval in colorectal cancer. However, some of the combinations, even though preclinically look incredible, have run into some difficulties. And what are they? Well, preclinically, one of the thoughts was, okay, well, how about in non-small cell lung, we combine it with EGFR inhibitors such as afatinib. Preclinically, it looked wonderful. However, from an efficacy standpoint, and the numbers here are relatively small, we did not see a significant increase in efficacy. But what we did see was a significant increase in toxicity. So our grade 3 event rate was close to 33%, a lot of diarrhea, a lot of severe rash. As to whether this combination will move forward is unlikely given the fact of this therapeutic window. Likewise, with MEK inhibitors, the data preclinically also looked beautiful. But when we got this into patients, the efficacy was not that much greater than single agent alone. And ultimately, the toxicities here, again, grade 3 toxicities, close to 34% of these patients may impede this from moving forward. One of the big questions, and questions that are still trying to understand, is how does KRAS inhibition affect the immune tumor microenvironment? This paper, this was published in the 100th anniversary of Nature. Dr. Cannon from Amgen, who developed the sotorasib molecule, published this data showing that indeed inhibition of KRAS G12C by sotorasib can create a pro-inflammatory environment, that in combination with PD-1, you can lead to significant suppression of tumor growth. However, we ran this part of the CodeBreak 101 trial where we combined this with atezo and also with pembrolizumab, and unfortunately has led to significant toxicities. In some doses, close to 100% of these patients were developing really grade 3, which is severe liver toxicity. It didn't matter if we tried to sequence the drugs, we were still seeing high rates of liver tox at close to 30% and above. And unfortunately, the response rates were not that much more than single agent alone. Now, ultimately, how this will pan out, whether in duration of response and PFS, is too early to tell. But right now, it seems somewhat difficult for us to now combine this with a checkpoint inhibitor, given the fact that if you're a lung doctor, you can give carbopem-pem and some would argue that, close to 30% of those patients will have a 5-year survival. I don't know if anybody knows this date, May 6, 1954. I'm a big history buff. In fact, I asked Anthony if I could meet Neil Ferguson when he was here, but he's not here. But do you guys know this date, May 6, 1954? Anybody? No. So this was the day that Roger Bannister broke the 4-minute mile. Now, it's interesting. Before Roger Bannister broke the 4-minute mile, there were experts from Harvard, not Stanford, Harvard and other places who said, this is humanly impossible. People will have their hearts burst. People are going to die. Roger Bannister, May 6, 1954, broke the 4-minute mile. And a month later, John Walker, his friend from New Zealand, broke his record. It was almost as if that 4-minute mile was a psychological barrier more than an actual physical barrier. After the development and the approval and the evidence that's showing that KRAS G12C worked, there has been an explosion of new therapies now targeting KRAS. It's as if that G12C inhibitor broke the 4-minute mile. The next mutant allele that is most likely to break, hopefully, is going to be G12D. And there are several G12D inhibitors in, last week I put the first patient on Mirati's KRAS G12D inhibitor, a pancreatic cancer patient, on this trial. I hope this thing works. We know preclinically that it's very selective and very effective. A drug that came out of nowhere is this molecule called, from Revolution Medicine called RM6236. This is a company here, right here in the Bay Area. And they hypothesized something that was not really that known widely in the literature, that activated RAS will bind to something called cyclophilin A, and then will then bind to downstream effectors like BRAF. So they said, why don't we develop a drug that binds to activated RAS, cyclophilin A, and the cyclophilin so that BRAF cannot bind. Preclinically, this drug looked better than any other RAS inhibitor. It is a pan-RAS inhibitor, right? So theoretically, it could target any mutant allele of RAS, but not only any mutant allele, any isoform of RAS. Now, what was concerning was the godfathers of RAS, Frank McCormick, Channing Durr, Martin Bartelli, would have told you, do not do this. People will die. There were embryonic mouse models that were with KRAS knocked out, and they were dying. They were not viable. And interestingly enough, there's not a whole lot of literature on what does wild-type RAS does in a normal human being. And so, you got to trust the godfathers of RAS. They were scared. But we went ahead, and Revolution went ahead, as all companies that don't care, went ahead and went to study. And interestingly enough, this drug has been incredibly well tolerated. One of the most least toxic drugs, oral drugs, that I've ever worked with. Very few nausea, vomiting, and they develop this acneiform rash. It's more called like milia, which is, when babies get these little white heads. It looks more like that. But what's fascinating now is that it is working. We hit 80 milligrams, and all of a sudden, patients started to have massive tumor shrinkage. And some of these, you see these arrows that are on the stable disease that are ongoing. Many of these patients are continuing to have tumor shrinkage. I wish George was here. Is George here? George's not here. This is his patient he's sent to Huntsman Cancer Center. You can see that large mass on that right. This patient is now almost in complete response. This is a pancreatic cancer patient. I have never seen a drug work like this in pancreatic cancer, okay? Many drugs have fallen at the very feet of pancreatic cancer and have died. This is incredible. Not too far behind Revolution Med is Mirati, and they are also developing a pan-RAS inhibitor, and we will have that molecule in our clinic in the near future. One incredibly exciting thing is this, and if some of you are involved in immunotherapy, mutations are oftentimes expressed in cancer cells as neoantigens. So these mutational proteins are then processed by the endoreticulum. Those bits and pieces are then united with HLA proteins, peptides, and expressed on the surface of cancer cells. Theoretically, an immune cell that has the right TCR to that HLA could attack and kill that cancer cell. This is not only in relationship to KRAS, this is relation to any mutation, P53, et cetera, et cetera. The principle of this was already shown by Dr. Rosenberg's group in I think this was in 2015. Eric Tran showed that you could do this with TILs. And already several companies, including Infinity, which we're going to be involved in, and also Genentech, which we're also going to be involved in, are developing TCR, T-cell directed therapy against KRAS. So why would you do that if you have a good small molecule? Well, it turns out, again, remember, with small molecules, we often see resistant mutations. But theoretically, these TCR, T-cell therapies, it would not matter if they develop resistant mutations. You could still target these cancers. And again, Dr. Rosenberg's group published this not too long ago, in June of 2022, the first neoantigen T-cell therapy against KRAS G12D. And as you can see, it works. This is our KRAS portfolio. Our hope is to, I think we have developed the largest repertoire of KRAS-directed therapies anywhere in the world. And our hope is we recognize that we are on the cusp of learning what these drugs do, but also learning from the patients, right? Every single patient is an experiment. If we can learn from their blood, from each biopsy that we take from them, every data point in toxicity, we will make strides faster than if we just kind of let this go. So in conclusion, if you're in the lab, I want to tell you something. What you do matters, because this is what happened 40 years ago. And I see drug development now, right now, because I don't have a lab. I don't run a lab, but I work with a lot of people in the lab. The pace of innovation from the lab to new drugs is getting faster and faster and faster. What took 40 years, I will not doubt you, will now take 10, if not 5. Although past therapies did not work, as you saw in the case with tipifarnib, we are learning. Because we learned the biology, we can reuse these drugs in the past in ways that we hadn't been able to do before. These G12C inhibitors have really been the mile breaker that helped us bring in new drugs, and particularly non-small cell lung cancer. And there seems to be a differential response across histologies, but we know from past stories, right, past examples such as BRAF, that we can augment that with combinations with EGFR. Understanding resistant mechanisms, both acquired and also pre-existing, will be key for us to really drive benefit in these KRAS patients. And lastly, we're at base camp 2 now, and you know, if you had told me as a fellow if we'd ever figure out a way to fully, you know, cure patients with KRAS, I'd probably say absolutely not. That's kind of crazy. What are you talking about? What are you smoking? but seeing this portfolio of drugs and new therapies that are emerging, I would not doubt that in my lifetime, my career, we will start curing these patients with KRAS. Okay, so I don't know if any of you guys watched this launch. This is the Super Heavy in Boca Chica, Texas. I've been to Boca Chica. It's like in the middle of nowhere. Great fishing though. And remember the Super Heavy went up and everybody was cheering, and then this thing couldn't dislodge, and then it went into a spiral, and then it exploded, right? But it was kind of odd, and both my son and I were watching this on YouTube. They were all like clapping after a while. Like everybody from SpaceX started to clap, right? Like, oh. And I think that attitude, that Silicon Valley attitude that Elon Musk brought to Texas is that, you know what? Even though this thing failed, we will learn, right? We will learn, and we will make it better. And we will eventually get this thing to Mars, right? And I think that is the attitude that we have to have in oncology drug development. There will be other KRAS failures, I guarantee you, but we will learn. And if we can learn from every patient is an experiment, we will be able to really reach that height of the summit of KRAS Everest. All right, that's the last slide. I want to acknowledge my patients and our caregivers, obviously. This is our investigational therapeutic group. I run what's called the CTRC also, and what's called our Precision Oncology Decision Support Group, and all my collaborators, both in the institution and out. And most importantly, I want to thank my clinical team. This is my clinical team that is made up of all these research data coordinators, because ultimately, if you're a research coordinator, you're the ones who are doing the heavy lifting, okay? I'm not. You guys are the ones that are doing the heavy lifting, so thank you for that. All right. And as in Texas we say, thanks all, y'all. Steve, question.
Very inspiring. So maybe I missed it. You talked about the fact that, let me back up. I mean, those of us who have studied RAS in the lab, like we know the preclinical literature, like the mouse models literature, those tumors that initiate from RAS are completely dependent on RAS, right?
Yeah.
RAS off, tumor melts away.
Yeah.
So that's what we would have loved.
Thought.
What we would have loved to have seen in the patients.
Yeah.
And so it's somewhat shocking to have 30%
Response rate.
And then you showed us the follow-up studies that showed
Co-alterations.
secondary
Alterations,
right,
Yeah.
or amplification or upregulation. But do we know in the patients who never respond, what exactly is happening to patients who never
Well, so Fernandez showed some of that data, like for some reason the KEAP1 co-alteration, SMARCA4, CDKN2A specifically somehow blunt the effect of these G12C inhibitors. These co-occurring mutations that occur along with KRAS G12C. Why exactly? I don't have the answer for you because we're still, I know Nando's is looking at that specifically. My guess is just like with EGFR, right? That it is an alternative kind of pathway, that human cancer, unlike in our very kind of sterilized in vivo models, are far more complicated. But we know SMARCA4, KEAP1, CDKN2A, and he did a further analysis. There's a cancer discovery paper that's going to come out for that there are other alterations, interesting, like mutations in NTRK that seem to co-locate to poor response, poor efficacy, et cetera. As to why those are the case, we don't have all the full answers at this.
In the sense that might mean that all these amazing second and third generation inhibitors might not be that much better because
maybe.
It's this inherent resistance.
Absolutely. Yes, sir.
Do changes in the tumor microenvironment differ between those who respond and those who don't respond?
That's a great question. I don't know the answer to that question. Yeah, I know. That's a great question, and that's why we have to get biopsies on every single patient who goes on these KRAS inhibitor studies, There was a paper last month, I think, and I didn't have time to put in here, that definitely shows that there is tumor microenvironment changes, at least preclinically, in syngeneic models where they show regulation of interferon and upregulation of interferon when you give G12C inhibitors, but we don't know. Yeah. Right.
Immune systems.
Right, right, exactly. So we don't know yet. That's why we are, those lists of protocols we say, we're creating something called the RAS Stride program, which essentially means every single patient I put on a KRAS directed therapy, we're going to get, there are usually research biopsies that we send to the sponsor, but we can take an extra pass and keep that. We can get extra blood on those patients. We can collect all the data, and we can figure out why these patients are behaving as they're behaving, right? And that opportunity is here. And we can do that. So that's our hope, and hopefully, be able to collaborate with our translational scientists at MD Anderson to really kind of tease out the details of that. I'm not a lab guy, but kind of understand some of the stuff that you guys do in the lab. But if I can get you samples and you can interrogate them and you can tell us what the next best combination or generation of drugs, awesome. Yes, sir.
Yeah, so I have a question about like tumor agnostic, because I know for any type of tumor agnostic approval, BRAF, first was in melanoma, non-small cell cancer,
Yeah.
but then when it's match became tumor agnostic. What is the benchmark for?
Yeah, so there's, by the way, I wrote a great Nature Medicine review article on tumor agnostic therapies. A lot of it, I think, has to do with the FDA. So the FDA, right, gave the first tumor agnostic therapy with checkpoint inhibitors in MSI-high patients. And if you look, I call the FDA, they're like the Supreme Court. Like, they rarely make any major changes, and they usually, if they rule a ruling like tumor agnostic, which was a total new ruling, right, they usually follow that same ruling, right? And in that ruling of MSI-high, they had at least 15 different tumor types. The response rates were around like 30%, right? And of those 15 tumor types, at least the 5 major tumor types are all represented, right? Like colorectal, et cetera, et cetera, et cetera. So my guess is that you'd have to follow that kind of same trajectory or show efficacy in that regard with these small molecules. And the same thing with NTRK and so forth. That almost followed the exact same pattern. Now, one thing I'll have to tell you, and this is just an understanding of drug development, now the FDA has become much more strict, okay? I don't know if you know this, accelerated approval now is not just single-arm studies. They want randomized trials, right? And so for whatever reason, they're becoming much more strict. And so I think you're gonna probably have to show a higher bar of tumor agnostic efficacy than it was in the past. And that's not because there's any scientific specific rule. It's just because they, Yes?
So I'm not familiar with the metrics used in clinical trial, but
Yeah.
for a patient that has advanced metastatic cancer,
Mhm.
they treat it with the KRAS inhibitor.
Yeah.
So do we see the same response in the primary tumor versus the metastatic tumor?
Yeah, that's a really good question. So in clinical trials, we have very blunt tools. Someday, when we have generative AI being able to do tumor volumetric analysis, maybe it will be much more accurate. But essentially, what we use is a criteria called RECIST. And they're just a set of rules, okay, that are picked out by either me or like a radiologist and says, okay, we're going to measure this as a target lesion, this is a target lesion. We don't always measure the primary lesion, okay? And then we say, we're going to follow those patients 2 months later and see if there's been a decrease of 20%, sorry, or 30% or more, or if there's been an increase of 20% or more in the overall diameter of those measurements. You say, who came up with these rules? And these rules, to be very frank with you, there's a whole page, and Chris and I can laugh about it. These rules were established many years ago, but they are essentially what we use to define things like response rates, progression-free survival, etc. The question that you have is a very interesting question. We don't know the answer to that question because a lot of these patients who have metastatic disease no longer have their primary, right? So pancreatic cancer patients oftentimes who present at a stage where they can get a Whipple, but they then have metastatic disease. They get their pancreas taken out and they get metastatic disease in the liver, et cetera. So there's no way for us to measure their pancreatic disease because they don't have it at this point, right? So that's a very interesting question. Is there a difference between the primary tumors versus the metastatic disease? Are there differential responses? That's all very good questions. It's just harder to do that in a clinical trial kind of way to measure. Okay, yes?
I'm fascinated by your toxicity result.
Yeah.
Right, and so I'm sort of wondering, if sort of comparing the two methods, it seems like one is sort of, we're going to hold you in the off position, prevent you from doing something you want to do. And in the other case, we're not going to replace the lamp. And so in terms of like RAS, we have sort of how the two different sort of approaches work.
Yeah.
And so I'm just curious about what does that say if one method is sort of saying that toxicity is incredibly low and the other is incredibly high.
Yeah.
That's saying something about those methods or inhibiting it in a way that tells us about sort of the purpose of that in the first place. Yeah,
that's a really good question. Well, concerning the RAS-on inhibitor Revolution Med, it's definitely a different toxicity profile. But it's still early. I mean, I'll be honest with you, we're still in dose escalation. We're at 160, we're going to go up to 300. Even though we're seeing responses already at this level, and these slides are all public, so you guys can pull that down. I'm not giving you stuff that's not already published, already reported. But that's a really good question. Toxicities are very good because I do my career on toxicities. Why there are differences, even when you look at adagrasib versus sotorasib, there are differences in toxicity, even though the molecules are very similar in how they function. For example, what I didn't show you was adagrasib can be combined with checkpoint inhibitors, unlike sotorasib. And why is that the case? My hypothesis is really, it's the PK characteristics. It's something that's not very sexy in phase 1. We do all these fancy PD biomarkers, but it's the PK. So if you look at sotorasib, it has a very high Cmax, and then it goes like this, right? And has a very short half-life. Adagrasib has a very long half-life. It's 24 hours. Now, Dan Cannon, the creator of sotorasib, said, it doesn't matter, these drugs are covalently bound, irreversible inhibitors that switch to pocket. The turnover time of KRAS protein is 22 hours. You dose them once a day, should not matter. But I think the human body is probably far more complex than either structural biology would allow or the in vivo models. And I think it has to do with this PK profile that probably lead to either drug accumulation or not drug accumulation in the liver relative to checkpoint inhibition. So lots of interesting questions. You must be a phase 1 doc since you're into now.
I'm a health policy.
Okay, you're a health policy. Well, you can come and join our clinic.
But it's not good. Thank you.
Yes. Any other questions? Yes.
Is there any evidence that dynamic EGFR activation could be a resistance mechanism in cancer types beyond colorectal cancer?
Yeah, that's a really good question. That is a really good question. My old chair, Dr. Rizal Kurzrock, after we published that data on vammarintinib and cetuximab, once said to me, I think, she said, this is probably not just in colorectal cancer. I bet you this is in other tumor types, David. Why don't you test it in other tumor types? I never got the funding to try to test it in other tumor types. I don't know the answer to that. I wouldn't be surprised. I wouldn't be surprised.
Because it's interesting, because in colorectal cancer, you can give EGFR inhibitors only for KRAS wild type.
Yeah.
So there's
Yeah, exactly.
The cell organ, one animal question on that.
Yeah.
There's a subpopulation of alveolar type 2 cells that have stem cell activity.
Really?
And the EGF receptor KRAS pathway
Yeah.
is a mitogenic signal pathway.
Okay.
So if you block that, you
Yeah.
slow or stop the proliferation, but aren't expected to kill the cell.
I see.
It's just
Yeah.
But there is a whole other pathway that selects those cells and provides them with stem cell activity.
Interesting.
So instead of thinking about the second drug just coming from another drug
Yeah.
on the EGF-RAS mutagen pathway,
Yeah.
it would be smart to target the other signaling pathway that controls the stemness.
Yeah, such as.
It's a Wnt signaling pathway.
Oh, yeah. You know what? Actually, that's something that we're actually working on. There was a screen that one of my colleagues at MD Anderson did showing exactly what you suggested, that Wnt signaling may really synergize with direct KRAS inhibition. So, you know, I'm not a lab guy. So I saw this. It's funny because I'd known John Shen very well, but I had no idea he was doing this research. And I saw his poster at ACR, and I said, John, you know, I work on KRAS. He was like, yeah, I do know, but I said, you never shared this with me. He goes, yeah, I just didn't have time. And I said, why don't we pitch this idea? Because I'm running a Wnt hedgehog inhibitor. Why don't we pitch this idea to Endeavor, and let's do this combination. So we're doing that right now. And in fact, we're developing some of the preclinical models, and we're going to ask Endeavor if they would want to fund a trial of their Wnt inhibitor plus EGFR. So I'm sorry, your name was, maybe I should let them know that. Yes. Dr. Resner, thank you. Grasno, yes.
But one of the challenges with Wnt pathway
Yes.
inhibitors is they're broad and they tend to
Yes.
and so the side effects of that.
Yeah, yeah.
But the new generation, including the ones that are being developed here at Stanford and
Yes.
are very selective for the
Yes.
specific sex
Yeah.
variants, within that.
That's great.
And so the idea would be to know the one that's specifically
Yes.
active in the stem cells that
Yes.
they rise and propagating.
That's great. Look at that. It's awesome.
Okay, I think we should thank David.
All right, thank you.